So thank you for understanding and welcome. Yeah, Chris mentioned already the title and that you will get the slides. The slides will also include all the links to the publications. So you don't have to Google for the papers. You can just click on the link in the PDF file there. Just a brief overview. Let me quickly share my screen because that's what I need to do. Okay. Okay, so here's the brief overview. I will talk briefly about what we know about microRNAs at this point. So if you're working already in the field, of course, that's a given. Then briefly about the timeline, some recent review papers about this field and about microRNA profiling also in general. Then on the current methods, I will mention the two most common methods, I would say, for overall profiling of all microRNAs, next-gen sequencing and microarrays. And then probably the most interesting part for you as researchers in the field will be the case studies, where I will highlight a couple of papers that have been published within the last 2 years, going from pure discovery all the way to biomarker development and therapeutic, potentially interesting microRNAs. And then, like Chris said, as Chris said, at the end we will try to answer as many questions as possible. Okay, so what we know currently about microRNAs is that they're small non-coding RNAs, of course, typically around 20-22 nucleotide longs, a little bit longer in the case of plants. But in general, I will focus, of course, here on the mammalian microRNAs in this talk. Then they exist as precursors that are processed, and these precursor molecules have a very well-defined structure and have a very strong secondary structure. So typically, under, for example, microarray conditions, under hype conditions, they don't unfold. They have a stem loop structure as well as a hairpin structure. They're highly conserved, so if you look at cross-species conservation, it's really remarkably high for a lot of them. They bind to complementary messenger RNA molecules and act as negative regulators of translation. There are 2 different mechanisms, depending on whether it's mammalian system or plant system. I will go into that in a little bit. And they are typically highly expressed. What you usually see is that you have a handful of microRNAs, no matter which tissue you're looking at or which fluid body fluid you're looking at. You have a handful of microRNAs that basically represent 50% of all the microRNA expression level. The others are expressed at lower levels. And of course, what makes them really interesting, especially for the biomarker development and also as targets, is that the expression is tissue and developmental and, of course, also disease specific. So the current snapshot is that we have more than 25,000 mature microRNAs. A total of almost 200 different species are cataloged in Nearbase, which is the publicly available database, which includes all known and validated sequences. Nearbase gets updated regularly, typically every 6 months. The latest update took a little bit longer, simply because the amount of sequence information was so big. But the latest update was in August. So all the profiling methods and so on, they typically refer to the Nearbase number to make clear which sequence information was used. So no matter whether you're talking about microarray content, probe content, or qPCR primers, the Nearbase is usually what is referred to. The mechanism of microRNAs is rather complex, because each microRNA can control multiple target genes, and the other way around, it's also true that the target gene can be regulated by multiple microRNAs, so it's rather complex. The current estimation is that roughly 1/3 of all human genes are being regulated by microRNAs. As mentioned earlier, there are two different mechanisms that are being discussed. One is predominantly present in plants, where you have complete pairing of the microRNA to the messenger RNA target. So translation is being repressed, and as a result, messenger RNA is being degraded. In the mammalian system, you have typically imperfect pairing. So you have binding that's tight enough to repress translation, but it doesn't trigger degradation of the messenger RNA yet. Looking at the history, everything starts in 1993 with Lin4. That was shown to encode 2 small RNA molecules, which was rather surprising because at the time it was always thought that RNA encodes for proteins. And then in 1998, RNA interference was observed for the first time, which then consequently led to the Nobel Prize in 2006. And yeah, it took quite a while, I would say, for the scientific community to get used to the idea of microRNAs, simply because it was something that was really challenging the central dogma of biology at the time, which didn't really have any place for an RNA molecule that was actually regulating the DNA transcription. So back then the thought process was that, or the accepted model was that DNA simply gets transcribed to RNA, and RNA doesn't do anything but translates into a protein. Then from 2000 on, everything went rather quickly. In 2007, LED7 was identified in humans and Drosophila. In 2001, 3 groups, Bartel, Tuschl, and Ambrose, discovered that there's a rather large class of small regulatory RNAs, and that's when the name microRNA was coined. The processing of microRNAs from primary microRNAs to precursor microRNAs to then finally the mature microRNAs was already discovered in 2002. And when you look at the development of the methods over time, it's of course arrays were adapted first in 2004 for the use of microRNA expression profiling. Of course, earlier qPCR was used, but if you want to look at the entire microRNA profile of a sample, microarrays were really the first option. So that happened in 2004. 2005 already, the first study was published using next-gen sequencing, and then 2006, with a dramatic increase of sequence knowledge in the field, MIR-base, the publicly available database, went online at the Sanger Institute, and now it's hosted by the Manchester University. 2008 then, the early samples were typically dealing with cell line samples or tissue samples. In 2008 then, the first publications showed up that were talking about circulating microRNAs. So in body fluids like serum, plasma, blood, etc. And that trend certainly continues nowadays. I think the latest is probably urine samples that are being looked at. A little bit more specific in looking into the cardiovascular field, it's certainly one of the fields that adopted pretty early on the microRNA field. Back in 2005, 2006 is pretty much when everything started. It initially started with the discovery of microRNA #1, that it's a discovery that's essential for proper muscle development in Drosophila initially. That's where it was found. And then this was then continued in 2006, role again of microRNA #1 and 133, as well as 2006, how they affect and how these muscle microRNAs regulate myoblast proliferation and differentiation in the skeletal muscle. And then the first really comprehensive publication was a PNAS paper by Eric Olsen's group and Eva van Roy as the first author. And that was in 2006. And this paper described the expression of microRNAs and as their involvement in regulation and response to cardiac muscle hypertrophy. So basically 2006 was the year where the clear link between microRNA and the cardiovascular field was made. Not going into much detail, but in 2008, 2009, then other additional microRNAs were discovered and how they fit into the field. 2010, then stem cell fate of cardiac stem cells was discussed, which microRNAs play a role there. And then as the entire field moves on from regular tissue samples to body fluids that can be, of course, used for very non-invasive diagnostic purposes. Also in this field, in the cardiovascular field, circular microRNAs were then described and their involvement in coronary artery disease and acute myocardial infarction. And I will actually briefly touch on the paper for both of these cases later. Just briefly, very simple generalized overview about the microRNA processing. Everything starts with a primary microRNA, which has the typical stem loop structure here, and then the hairpin. And as you can see, it still has the 5' cap and the poly A tail at the 3' end. This gets processed by enzymes complex called DROSHA and PASHA to the so-called precursor microRNA. Basically, what happens here is that the 5' and poly A tail are being cleaved off. And this precursor microRNA at this point in the mammalian system gets transferred into the cytosol. And the cytosol then, another complex called the dicer molecule, is processing the precursor microRNA in a way that the remaining tails, 3' and 5' end, get cleaved off, as well as the hairpin structure gets removed. So what you end up is with the microRNA sense-antisense duplex. And this then can dissociate, and at this stage we talk about the mature microRNA. What is shown here is microRNA star, or with the asterisk duplex, is, like I said, the microRNA antisense duplex. The bulge here indicates that it's, of course, a mismatch that's typically present. So these are not perfect matches. This asterisk nomenclature has been retired recently with MirBase version 18, 19. So nowadays we differentiate these two by adding the ending -3p or -5p to the naming. If you have any questions about the nomenclature, I can highly recommend the blog, which you can find at the MirBase website. They have a link. The link is here. Like I said, in the PDF file, that will be also clickable. But if you just go to mirbase.org on the blog section, there's an article, a blog post called "What's in the Name?" And Sam from MirBase does an excellent job in explaining not only the current status of the nomenclature, but also the reasoning behind it. So if you look at the current the sequence knowledge for microRNAs that's available so far, this is a brief overview showing you the almost exponential increase. This lists the number of entries in MirBase from version 6.1 to version 19, which was, like I said, just in August. And you can also clearly see here the effect of that next-gen sequencing had on the field. The impact is quite remarkable. So 2007, 2008, that's when Illumina released the first small RNA sample prep kit, and you see since then you've seen a dramatic increase in sequence knowledge. And of course, accordingly, if you look at the number of publications in PubMed that that deal with microRNAs, it's also pretty much an exponential increase. If you fit the curve, it's really impressive. Of course, for 2012, that's just projected, but basically it's going from 2001, basically within 11 years, from almost nothing to more than 6,000 publications. So what makes microRNA so special in the cardiovascular system, or why you could be interested in it? There's a whole variety of reasons, of course. Some are specific to the field, others are more general. One, of course, is that several microRNAs are specifically expressed and highly enriched in skeletal or cardiac muscle tissue. This group is typically called the muscle microRNAs, of course. And with that hand in hand goes that microRNAs are really essential for proper muscle development and exert post-transcriptional control during biogenesis. These muscle microRNAs regulate myoblast proliferation and are also responsible for regulation of the differentiation in the skeletal muscle. And I mentioned earlier already diagnostic potential. Circulating microRNAs might be used or can be used already as novel biomarkers for different diseases such as coronary artery disease, CAD, or acute myocardial infarction as well. Of course, dysregulation of microRNAs can also contribute to cardiovascular related diseases and disorders, the same as mentioned above, coronary artery disease and so on. And microRNAs are likely associated with other muscle related diseases. And of course, the most promising is that they are not only potential diagnostic targets, but potential therapeutic targets. So if we look at the potential for the different fields, microRNAs, of course, what you do in the diagnostic field or biomarker field, of course, you try to find, you do differential expression profiling of all microRNAs and try to find whether any particular microRNAs are significantly up or down regulated. Initially, it was thought that maybe one or two microRNAs can be used to establish this certain biomarkers, but as it turns out over time, you really have to look at a more than just a handful of microRNAs. Typically, you have to look at an entire signature profile, which usually consists of roughly, ballpark figure, 15 different microRNAs that you have to look at. And of course, one point would be to use to look at microRNAs in body fluids as biomarkers. There's of course work being done already there, and that would allow then for very accurate and detailed clinical diagnosis. There are certainly fields, for example, the cancer field, that is already very far ahead in this particular point. And what it's potentially also be useful for is looking at these signature profiles is to be able to determine the prognosis of a patient and predict the treatment efficacy. I just mentioned that the cancer field is already far ahead when it comes to the clinical diagnostic part. It's actually since this year there's a diagnostic kit commercially available, which is manufactured by Rosetta Genomics. It's called MearView, and it allows you to differentiate between the 4 different subtypes of lung cancer. The diagnosis is really extremely accurate, around 80 to 90%, whereas before it was around 50%. So that's a huge help for the clinician, and yeah, allows to pick the right treatment. Then when we go to the next step, drug development and therapeutics, there are microRNAs, of course, could be used for vascular regulating drugs, as well as simply as drug targets in cardiovascular diseases. And then, of course, the third point would be in general to study microRNAs to understand the response to to certain stress conditions and infection or other stimuli such as environmental stress, for example. If we look at a couple of review papers, this one was one that certainly stands out. It's a very comprehensive paper. Of course, it's from Eric Olson's lab. Was published in 2010. It was published in Circulation, and this is one of the main graphs from the paper, and shows nicely the different microRNAs that were identified. They're all listed here and how they interact with particular targets. And when you look here at this graph, you see an area that's green. In these areas, these microRNAs indicate stimulation of the target, whereas where the purple areas indicate repression of the target. And you also see the up and down arrows here right next to the names. That means that the microRNA is either up or down regulated in this particular case. You will also see some question marks here. These are in positions where there were initial reports about which were linking a particular microRNA to a target, but either there were not conclusive evidence in follow-up papers, or there's just still the need for additional validation. So, like I said in the PDF file that you will see, if you can just click on the link here where it says article, and it takes you to the right site. So this is a very good general overview paper dealing with microRNAs in the different tissue types in the cardiovascular field and their involvement in different diseases and functions. Then if we go to this year, in 2012, this particular paper here by Zhu and Fan was published in Cardiovascular Disease, and it's a very interesting and good addition to the previous paper, because it focuses really on the microRNAs that are involved in the extracellular system, basically the circulating microRNAs, and their potential role in cardiovascular diseases. So that this covers basically everything from blood, serum, microvesicle microRNAs, exosome microRNAs, etc. So this is another very good paper. And then the third one, again, focusing on circulating microRNAs, also from this year by the group around Zeng. It's not only focusing on the circulating microRNAs, but it's also looking at the potential for novel and to their use as novel and sensitive biomarkers. So this is certainly also of interest. And then a paper that I haven't had the chance yet to read is just came out in November of this year. It's a Nature review paper by Eva van Rooij and Eric Olson, and it talks about the microRNAs as therapeutics for cardiovascular disease, the opportunities and the obstacles that they are facing. Eric Olson's group had spun off, or he's co-founder of Merigen, one of the companies that's working on therapeutic in the cardiovascular disease. So I would expect that this is a really highly interesting article. Then if you look at the field in general, in terms of, especially if you're new to the microRNA field, this is receives my highest recommendations. This is a Nature review paper which came out in May of this year. It basically talks about the different microRNA profiling methods, mostly focusing on qPCR, microarrays, and rna-seq, basically outlining and highlighting the strengths and weaknesses of each method and what you should consider when designing your experiment. It also goes into detail in which method to use and how you extract your RNA depending on the sample type, your starting material. And like I said, it really helps to guide you through the process of finding the right method for your experiment. Oftentimes we see it, because we offer all three methods as a service, we oftentimes see people wanting to do next-gen sequencing, although it doesn't really make much sense in their particular case. So, and this is really a nice flowchart that helps you guide you through the process. Then if you look at the tools available for the different steps of microRNA research, first step, of course, is identification. Clearly, what is dominant these days is next-gen sequencing. There's no doubt about that. There are also computational strategies, of course, which typically will look at precursor molecules. And then what we've done a lot in the early years when MirBase was still young, so 2006, 2005, 2007, in those years, we did a lot of tiling arrays where we basically validated predicted sequences. And then when it comes to measuring the actual profile, of course, in the early days, Northern blotting was used a lot, and in situ hybridization. But clearly, the three prominent methods these days is real-time PCR, if you want to look at only a few select microRNAs. Of course, there are also entire plates, but you always it never covers the entire MirBase collection of all known microRNAs. You're always looking at a subset there. So we typically use that for validation of results that we generate with either arrays or next-gen sequencing. Then, yeah, array analysis, of course, is still the dominant method, and that makes also sense. Next-gen sequencing can be, of course, used also for profiling. But at this point in time, we would still prefer microarrays for the actual profiling and use next-gen sequencing only for the discovery step. And why this is, I will mention in a little bit. Target determination, of course, is then the next step. Nowadays, most papers include this step to a certain degree. Of course, you can use bioinformatic tools. There's a whole variety of databases out there. It's of course not easy because the binding doesn't require a perfect match of the microRNA to the messenger RNA target sequence. So in the mammalian system, you have really the problem there as compared to the plant system where you have a perfect match typically. Of course, you can look at gene and proteome expression analysis. This is sometimes what we do for customers is basically run the same total RNA sample on one hand using microarrays for microRNA profiling as well as messenger RNA arrays or sequencing. And in the plant field in particular, of course, it's because you have the perfect match, you can use degradome sequencing, which is becoming more and more popular. Then when it comes to the pathway analysis, there are a couple of free sites available that try to bundle up basically the information that's available across the web. Three of them are mentioned here. One is MirFocus, the other one is MirPath, and then MMIA. There's also a commercial solution by Ingenuity, which does basically the same thing. The difference is really that the commercial version is keeping, of course, a better track of recent advances than the free versions. Yeah, and then the final step to perform functional analysis, you can use luciferase assays. Then, of course, typically knockout models. You can inhibit your microRNA expression using antagomeres, or you can reinstate microRNA expression basically using microRNA mimicry. There are pretty much three basic steps involved. One is, of course, the discovery, then the profiling, and then the validation. Traditionally, as mentioned, this was done profiling with a microarray, and then those results get validated with qPCR. Or nowadays, that there's more sequence information available, some people feel comfortable to just look at a subset of microRNAs and do only the qPCR. And of course, next-gen sequencing is dominating the discovery field, the discovery step. And what we do a lot in not as well characterized species, no matter whether animal models or plant models. For example, the Chinese hamster model, there was only a single microRNA known for Chinese hamster until very, very recently. So in those cases, we could, or what we do is we pool samples from different developmental stages, do the sequencing for the pure discovery purpose of this workflow here, and then based on these sequence information that we receive from these samples, we create then custom arrays and do the actual profiling using the arrays, which is typically much more cost-effective and also faster. So, and we call this combination of the strength of both methods seq array. So, and these are the two methods that I want to focus on now. But before we go there, of course, we need to have decent samples. There's a whole lot of kits out there by different manufacturers that we can recommend, depending on what type of starting material you have. No matter which kit you pick, make sure that it's really one that's retaining your microRNA. There are some so-called total RNA kits out there that don't actually retain the microRNA, so you have to be careful there. Then the other thing as well, they all work. They will give you a slightly different profile. So no matter which kit you pick or which extraction method you pick, make sure that you stick to it throughout the entire experiment. Three good choices are mentioned here. Total RNA extraction kit by a Canadian company called Norgem Biotech. This is kind of unique because it's phenol-free. QIAGEN's offering is the so-called miRNA Easy Kit. Make sure which one you're picking here if you go with QIAGEN, because they also have an RN Easy Kit, which does not retain the microRNAs. And then Ambien has a kit that's called Nirvana. Typically, you need depending on the sample, on the profiling method, you need 1 to 4 microgram of total RNA. Then, if you want to estimate the quality of your sample, you can look. The easiest is really to use a nanodrop and look at the UV vis spectrum. So we look at the ratio of 260 to 230. That should be above 1, and the 260 to 280 ratio should be above 1.8. All these criteria that are listed here on the slide are really only applicable for regular average cell line or tissue samples. As soon as you go to, let's say, blood, plasma, serum samples, or urine, you really can't rely on any of these numbers here. It really boils down to trial and error at that point. Shown here on the right is a bioanalyzer run. Occasionally we do run bioanalyzer gels, but not routinely, simply because it doesn't really give you any good information about the microRNA content and the quality of the microRNAs. Of course, you can estimate the overall quality based on the quality of the larger RNA, but we had also cases where we had RIN values of, let's say, 3, so meaning the larger RNA was completely degraded, but the small RNA fraction was just fine. These are just 3 gels that were kindly provided by Norgem Biotech, which show basically different types of gels: formaldehyde agarose gel, Agilance bioanalyzer gel, and the urea page gel, and always showing good microRNA retention recovery of a kit on the left side versus no the missing microRNA fraction on the right side. Looking at the two different methods, arrays versus sequencing. Of course, when sequencing first became more popular, it was there was a lot of hype that that microarrays will go away, and it's a method of the past. But it turns out that arrays are actually still quite useful, and so they are around for a while, we think. The main advantages of arrays is, of course, it's very straightforward. It's a high throughput method. It's pretty robust. It's around for a while. Can be automated. Turnaround time is pretty short if you send it in your sample somewhere, or if you do it yourself, of course, that's the case too. And besides the lower cost, it's also simply the fact that the result, the data analysis, is really straightforward and easy to understand, especially if you're new to the field. The disadvantage, of course, is that you can use them only to look at known sequences, known microRNA sequences. So as long as you're sticking to well-characterized species such as human, mouse, rat, etc., any of the 193 known species in Mirbase currently, then chances are the microarray is the way to go when you want to do overall profiling of all microRNAs in the sample. The advantage of sequencing, obviously, is that you don't need to have to know, you don't have to have any knowledge about the sequences prior to doing the experiment. But not only that, you can look at different alternative forms like isoforms of the microRNAs, and depending on how you set up the sequencing, you can also look at other small non-coding RNAs and even messenger RNA. And of course, another big difference is that you're getting absolute numbers, basically absolute copy numbers versus relative values, expression levels, and arrays. Let's briefly focus on the arrays. This is just a typical workflow for us and probably for everybody else as well. First you do sample QC, then sample prep, and the nice thing here about the sample prep is basically you're using the total RNA sample straight. You don't do any amplification or any of there's no step involved that could really severely bias your profile or favor any particular microRNA over another. Then you do the high correction to do the data analysis, etc. So this typically looks like something like this. You run an experiment with a couple of biological repeats, run the array. What is shown here is as the first heat map is basically an overall view of all microRNAs and their expression levels, green representing that the microRNA is downregulated, red means upregulated. And in a case where you're just comparing two groups, like in this case where you're comparing control versus treated, you then would apply something like a t-test to compare these groups, and here are two resulting heat maps shown with different p-value cutoffs. So p-value representing the statistical significance of your data. And then if we switch to the sequencing side of things, the workflow is pretty much the same. The main difference is really that you're not using the total RNA as direct as for the arrays. The library preparation step is is rather involved, and there's a lot of discussion about the fact that there are actually so many steps involved, and amplification steps in particular, that you might be biasing your microRNA profiles at this step. And this is actually the main reason why if you want to just look at the microRNA profiling of different samples, we would still prefer the arrays. We have simply the opinion and the feel that it's that you're avoiding any potential bias to that you might be introducing through a library prep stage here in the sequencing field. So other than that, of course, then you have sequencing and then the data analysis as well. And just to give you some numbers that you can expect when doing sequencing of microRNAs, the machine that we're using for microRNA sequencing in particular is an Illumina system called Genome Analyzer GA2X. Read length is typically 35 bases, and the sample quality, of course, determines the number of reads, but typically we see 20 to 30 million reads per lane. The number of reads, of course, per sample depends on how many samples you want to barcode into one lane. So you can run multiple samples in one lane, but of course, as shown in the table here, you have the 30 million reads maximum. Typically, that's the constant, and then you have to divide the 30 million divided by the number of samples in that particular lane, and then this gives you the overview of how many reads per sample you can expect. And because you end up with only 10 million or 7.5 million reads, if you pick 3 to 4 samples per lane, this is really the maximum that we would recommend to run for any profiling. For just discovery purposes, we would probably run just a single sample per lane.
Christoph, if I could interrupt here just for a second. We're going to try something new today just to try to get listeners involved here, make this webinar a little bit interactive. So we're going to go ahead and launch a quick poll right now. And since we're talking about RNA-seq and we're talking about conducting experiments looking at microRNA, we want to ask the question, generally speaking, how many biological replicates from each test group are required for good RNA-seq data? Now, this is going to depend on the type of species, type of sample. Let's assume that it's a human tissue sample and you've got multiple different test groups and you want to look at the expression across those groups. If everybody could just quickly just pick one, we'll leave it open here for another 30 seconds or so, and then we will show you the results in just a few minutes later on in the webinar. And we're getting very close. We've got about 60–70% voted. Give you just another minute. Okay, we'll go ahead and close it. Now give me just 1 second and I will share the results with you.
Do you want to share the result first?
Yeah, let me share the results first. Okay, what do we have? 47% say 3 replicates. Great.
You need to close it. Yeah. Okay, great. Thanks, Chris. That was actually interesting because we see a lot of times that people think that only because they're using RNA-seq, a fairly advanced method, that they don't need any biological repeats any longer. And yeah, so that's great to see that everybody was on board.
And that nobody selected just one replicate, because as we know, there is if you have one data point, you have 0 data points.
So that basically, I can skip this slide here then, but obviously I don't have to make the case for biological repeats. So yeah, 3 is basically the minimum that we would recommend for regular cell line or tissue samples, or any type of animal model. Typically that's sufficient to get some good initial data, and to be able to make a call then when it's when you're moving on to clinical samples, human tissue samples, then that's a completely different story. Then it will depends, but anywhere between 10 to 30 samples is a good start. We actually recently had trouble There was one publication because 30 samples per group were not considered to be enough. So, but there then will depends on, of course, also the data, how clear they are. Yeah, this was just something that came up all the time with potential customers or customers, and so that's why we thought it was interesting to do this quick call here. So when we continue the in the sequencing workflow here, when it comes to data analysis, this just as a brief overview what our software does. Basically, we're taking, of course, what's commonly done is you take the raw reads and you convert them into mappable reads. You try to map them to the known mammalian microRNAs on near base, and then or you try to map them to messenger RNA, etc., so that you can throw them out. And you basically go through these different mapping processes here, and then you end up pretty much with 3 different groups once you filled out all the junk or unrelated sequences. Group number 1, of course, is the known microRNAs that you can map to near base. Then other potential candidate microRNAs, but that, let's say, map to other species. And then, of course, number 4 here is the most interesting one, typically for you when you want to do discovery. These are potentially novel microRNAs that haven't been described for any species yet. And just to give you another sample of data that you typically get from sequencing, and information that you don't get with any of the other methods is the information about the actual exact sequence and sequence length, the so-called isomer forms. So it's typically the case that you have one particular form of microRNA sequence, which is then listed in MirBase, which would be here basically the first sequence here, with almost 60,000 counts or copy numbers that were measured here. But besides this main dominant sequence, you still have other sequences, so-called isoforms. These can be truncations at one of the ends, like the second one here. It's still pretty strong, almost 11,000 reads for this particular sequence, or truncated at the other end, or sometimes shifted, or even single-point mutations, as in the bottom 2 cases. And there's a lot of discussion still going on about how much of this is actually real, how much is introduced during the library prep stages of the sequencing experiment. But I think its overall agreement is that they are isoforms. So I would say the top numbers here are probably real. And yeah, so this is information that you, of course, can't get with qPCR or with arrays. An array would not be able to differentiate between these 2 sequences here at the very top. And that's, of course, potentially one of the main reasons why you sometimes have discrepancy between your array data versus qPCR data. Because with qPCR, you're looking really only at one of the 2 sequences here in this particular case, versus the array that will give you the sum of both. Okay, that's so much as a brief overview over the methods, and now a couple of papers that highlight basically the application of these methods as they apply to the discovery, biomarker, and therapeutic field. So this first paper here was done using sequencing, and it basically it's a discovery paper. It describes, characterizes and describes the discovery of novel microRNAs and so-called mRNAs in mutated SC2 cells. What mRNAs are basically microRNA offset RNAs. It was frankly the first time that I read this in this paper, about those in this paper as well. What they've done is basically sequencing of these samples, and they found 652 mature microRNAs, of which 21 were highly expressed. And what they looked at then in particular was the distribution of the different isoforms, something that I just mentioned in the previous slide. So, and that's basically what's shown here in this graph. It shows you the expression of the different particular microRNAs and how they basically how many isoforms they found for each one. For example, for the first one here, HSA microRNA 2.1, they found predominantly 2 isoforms, the black one and the blue one here. And then the white areas here are sequence variants that are not classified as isomers. So these are other mutations. And you can see here these some consist only of one isoform here, like microRNA 92A, and others of 2 almost equal one, like here microRNA 17. So the bottom line of this paper was basically that the majority of microRNAs were a mixture of sequence variants. And another interesting thing was that really the sense and antisense, or formerly microRNA and microRNA with the asterisks, or nowadays microRNA-3P and -5P, were pretty much found to be at the same expression level, 50-50. So that was another interesting result, because initially a lot of people thought that only one of the 2 mature microRNAs will be actually functional. So this as a basic discovery paper that just came out in 2012 in Blood. This was basically a paper that highlighted the complexity of microRNA mediated gene regulation in these particular SE2 cells, and forms the basis for future studies in this field. And if we move on to stay in the discovery field, but switch the methods. Basically, this is a paper from 2008. Isha von Roys and in Eric Olson's group, a pretty comprehensive PNAS paper again. It looked at acute myocardial infarction and how microRNA regulation is involved. And they used microRNA arrays, as mentioned, and validating these results then with qPCR and also with northern blot. The result was that the microRNA 29 family was found to be of high importance. And the conclusion was basically that microRNA 29 acts as a regulator of cardiac fibrosis and is a potential therapeutic target. And this is exactly the time when Eric Olson co-founded Miragen then. So, and what you see here in the graphics is basically the sham probe control heart, then the 3-day post-myocardial infection, and then 14-day post-myocardial infection. And you see how the heart structure suffers, and basically it loses the cardiac contractility. And the results here in B summarizes the microradiator, and in C shows then the validation basically of select microRNAs that were found to be up and down regulated in the arrays. Those were then validated using qPCR here. And then in E, it also shows you the actual northern blot analysis. So this was one of the first really comprehensive papers in the cardiovascular field, for sure, and important for the whole discovery of the involvement of the microRNA 29 family. Then when you go further into the diagnostic field, this is an interesting paper which was published by Johnson Godwin in 2010, again in PNAS. And it basically identified the biomarker or the microRNA signature of renal ischemia reperfusion injury, or short IRI. And method of choice back then was again microarray expression profiling. Again, of course, a sham control probe, naive samples, and also, of course, IRI affected ones. And it was shown here. The A shows the heat map of select microRNAs results from the microarray. And what you see here is the time course, basically a naive, then day 1 to all the way to day 30. And in B, what's shown here in section B is basically just the mean intensities for these particular microRNAs and the samples. And the main result here was basically that a signature pattern could be established. And so basically this microRNA profile here was these select microRNAs that are shown here represent the molecular fingerprint of renal injury. And yeah, so this was the accomplishment of this paper. In general, they could be basically clearly identified in the different categories. Then if we go further in the diagnostic field and move from cell tissue samples to body fluids, this is also a paper from 2010. It's dealing with the peripheral blood microRNA signatures and whether they can be utilized to identify patients exhibiting atherosclerotic CAD, coronary artery disease, of course. And this paper is actually based on qPCR data. So qPCR was used for the profiling step, and the result was that the presence of coronary artery disease generally coincides with a 5-fold increase in the expression levels of microRNA 135A. That's shown here in figure B. And then a 4-fold decrease of microRNA 147 at the same time as compared to the control. So this is shown here in B. And the problem that you have with qPCR data, typically when you do profiling in tissue, it's fine. There are some sequences known that you can use as controls. When it comes to blood samples, on the other hand, and plasma and serum, things become more tricky because there is really no housekeeping microRNA. Whereas if you're looking at messenger RNA profiling, of course there are housekeeping genes that you can use, but there is nothing analog in the microRNA field. So that's why I always cringe when I see here this housekeeping, although it's in parentheses, but it says here housekeeping microRNAs. The problem is that even a lot of people use MIR16, 16A or MIR21 for normalization purposes in qPCR experiments that are dealing with blood or plasma samples, but we've seen plenty of cases where it's clear that you can't really do that. This is an assumption that might simply not be true. So this is really one of the big issues when you do qPCR on these particular samples, body fluid samples. There's really still a big room for improvement to find a good sequence for normalization. Another approach, of course, would be to use just global normalization, and there are attempts to do that in the qPCR field as well. Then if you look at the potential therapeutic function of microRNAs, this was an interesting paper that came out also in 2010. It discusses the role of microRNA 210 as a target for treatment of ischemic heart disease. It was published in Circulation. Again, this one was done using microarrays, and the result was that it showed that microRNA 2010 was highly expressed in live mouse HL1 cardiomyocytes, and compared that then to apoptotic cells 48 hours after the hypoxia exposure. And again, of course, it was compared to also a sham probe. And what was interesting then, the therapeutic step was then basically what happens if you reinstate the expression levels of microRNA 210. So adult mice were injected with minicircle vectors carrying this microRNA 210 precursor, and the interesting result was that 8 weeks later, these mice showed significant improvement of the left ventricular fractional shortening in the minicircle vector carrying microRNA 210 precursor group, versus compared to the group that had just the microRNA scramble control. So this was one of the first papers really showing the potential therapeutic effect that microRNA, reinstating microRNA expression levels can have in the cardiovascular field. And then another paper in the same field is talking about microRNA 24 and how it regulates vascularity after myocardial infarction. Is microRNA 2024 is enriched in cardiac endothelial cells and significantly upregulated after cardiac ischemia. And this paper basically summarizes the finding that microRNA 24 acts as a critical regulator of endothelial cell apoptosis and angiogenesis, and that it can be potentially used for therapeutic intervention. It's basically the similar experiment as the previous paper. They tried to they used an antagomere 24 as well as a scrambled antagomere, and study the effects that each of those had. And the last example here is focusing on microRNA 29 and the downregulation of miR29 by antisense inhibitors and how it protects against myocardial ischemia reperfusion injury, IRI. And again, the method of choice here were microarrays. So this paper was published in 2010, and it was found using the microarrays profiling that again the microRNA family 29 was involved in important particular 29A and C, and it showed that the levels of 29A and C decreased remarkably after a 7-day treatment with PIO, and PIO stands for pioglitazone, which is a peroxisome proliferator activated receptor, and protects against myocardial ischemia reperfusion IR injury. And yeah, in the figures they just show again here. In, for example, figure A shows summarizes the array results for 29A. It shows the relative expression levels of the control, and then how the expression levels change based on the injection of antagomere to 29A or missense antagomeres. Okay, so this as a brief overview over some case studies. I hope that gave you a good overview and at least some ideas about the field and where we are. If you're new to the microRNA field, you might not have heard of us as LC Sciences. So we're really a microRNA-centric company, which is focused initially on our own microfluidic chip technology, which allows us to customize microarrays. So as a result, this allowed us to basically be the first microRNA microarray service provider back in 2005. And but in the meantime, we also do sequencing and offer as a convenience also qPCR validation. Yeah, we're located in Houston, Texas, so all the experiments are run here. And but yeah, we have customers worldwide and representations in certain parts of the world. And if you want to look at further papers, I can recommend also to just go to our website. On the publications, we have in the meantime more than roughly 600 customer publications, both sequencing as well as array publications, and they are sorted based on the field. So you can easily just scroll down to the cardiovascular publications and see what has been done already in the field. Yeah, this is just showing where we have representations. You will see Europe is missing simply because European customers, there's typically not an issue with shipping samples, so they just FedEx or UPS the samples directly to Houston. So I hope this was helpful. I will see that I can answer some questions now, but in any case, feel always free to email us at service@lcsciences.com with any additional questions that might come up, or give us a call.